Hey, everybody. Thanks for joining today. I'm excited to discuss with you guys about personalized medicine, personalized testing. It is intended to give you a good overall synopsis on what personalized medicine is and how it can benefit your clinic and just all the things that it can do for your patients. But I wanted to share a little bit about why am I in a room here or on a Zoom meeting with Dr. Jasby and you guys? Some of you might be familiar with me because of what I've done in ozone and my passion in that. Well, I met Dr. Jasby through a friend, I think about a year and a half ago now, maybe
Yeah.
even a little bit longer. But Dr. Jasby was doing a presentation and, still a scientist at ASU. And he just had blown me away. I sent him an email immediately afterwards and was like, hey, you got a really, really cool thing here. It's going to save a lot of lives. It's going to help a lot of people. And just, I think this is a great calling for you. And, in my family, cancer has been pervasive. I mean, my uncle, two grandparents, my brother have all passed away from cancer. And there's more than that, but that's just to name a few. And, my brother was really impactful to me. The fact that we lost my brother to cancer, that was a long journey. That was years and years. And thankfully, he lived much, much longer than the practitioners anticipated by I can't remember exactly, but I want to say about 16 years or so, something wild. And, we have been doing a lot of different things, both conventional and integrative. So hyperbaric oxygen was really helpful for him, got into ozone and all this stuff. But, when I was sitting across listening to Dr. Jasby speak, I was thinking to myself, like, if we had a test like this early on, what kind of difference would that have made in Matt's life? To be able to understand what's happening in the body before the symptoms appear, to have really good personalized protocols that give a really good idea on what to do about the situation that somebody is in. So I was totally sold exclusively on the idea of like, hey, we can use this to save lives just through early detection. And I think Dr. Jasby, you told me in the past that if we find things early on, the outcome greatly improves regardless of if they're doing conventional treatments or not. And as I understand, a part of that is because people have a psychological change. They have a change in perspective of how to approach their lifestyle, and therefore can change their outcome at a much earlier stage where it's much easier than if we're waiting for later stages. So if we can get early detection coupled with personalized protocols, it's just going to be a huge literal lifesaver in this world full of chronic disease. So, but I also want to let guys know as well. I mean, this is a business. This is Therium. I can't think of anything anybody more qualified to talk on personalized medicine than Dr. Jasby. But that being said, we do have a product, and I hope by the end of this that you do find it valuable and it would be something that you want to invest in for your clinic. That being said, there's no expectations on that. So we're here to provide value and information. And, regardless of where you go in the world of personalized medicine, I know listening to Dr. Jasby and hearing his wisdom and knowledge has always been beneficial to people, especially me.
I really appreciate that introduction, a very warm and heartfelt introduction. Like I'm so thankful and have benefited so much from your friendship as well as your involvement in Therium and our direction going forward. You mentioned your brother, and I always think that is the gold standard. That's the ultimate goal of any health testing entity, should be to establish a means or a mechanism by which routine monitoring can be done relatively inexpensively, accurately, relatively non-invasively, if at all possible. And there's various, criteria when we talk about routine monitoring. It really is the gold standard of healthcare, and not something that any healthcare system thus far has been able to get right. And so, far aside from just the Aristotle test, there's so many things that have to get done for us to get to that space. And as you mentioned, today we're going to discuss, various aspects of precision medicine. There is personalized medicine, there's precision medicine, they're often used interchangeably. We are talking today mostly about precision medicine, and we'll discuss maybe why that is different. And we're going to discuss precision medicine in the context of Alzheimer's disease, both in the detection of Alzheimer's disease, as well as the generation of a personalized or bespoke protocol to treat Alzheimer's disease and how that's done. And we're going to discuss two of my own publications regarding this. And so the title of today's talk is Revolutionizing Healthcare. And this is what it will take to get to a point in time where routine monitoring is taken for granted and isn't just the goal, but it is so routine and commonplace that it's taken for granted. And the title is Personalized Medicine, and we're going to discuss data collection and data analysis specifically. Briefly, I want to introduce a small portion of our science team. Of course, we have Dr. Ortega Santos, which is our personalized health liaison. We also have my co-founder, Dr. Alex Moore, and our chief science advisor, Professor Klein Sitharalman, professor at ASU's School of Molecular Sciences. And of course, myself, and we'll discuss some of my work today. As I mentioned, we'll mostly be discussing two of my publications related to Alzheimer's and the treatment of Alzheimer's. So really quickly, I want to discuss precision medicine, and I want to discuss what it is and why it is so very important. Really, it is the understanding of each patient's unique health profile. These are tailored strategies focused on individuals. They're based on genetic, environmental, and lifestyle factors. They are often unique to that individual. And analyzing an individual's data through these lenses, a systems biology approach to data analysis, is relatively new in the science. And really, we are still stuck in a currently dilapidated diagnostic model whereby we conduct these really disparate health assessments that are non-sensitive and non-specific. Therefore, we are not able to create tests that can differentiate closely related disease mechanisms, nor are we able to create tests that are sensitive enough to detect early characteristic shifts in biological functioning that often belie cardiovascular disease, neurodegenerative disorders, as we'll see today, cancers, etc. And so we are trying to set a stage here to discuss today the role of technology in enabling precision medicine. And so we've gone from these very traditional approaches whereby we order, a CBC and O test, and then that sort of narrows it down further, and then some further testing. And really, the reason why we want to get sensitivity and specificity in our testing is because we can actually affect treatments, whether we discuss therapeutic targeted interventions early on. As Micah said, these individuals have a much greater chance of surviving. That's true across breast cancer, Alzheimer's disease, cardiovascular disorders, and ultimately our idea, our enhanced efficacy has been shown that these targeted precision approaches have greater patient outcomes than the sort of traditional standard of care one-size-fits-all approaches. And how do we tailor that, we'll discuss later today as well. So our objective, of course, is to explore today how medicine is revolutionizing healthcare through this lens. So this is on the graphic you can see, and this is from an upcoming paper of ours we submitted to Genome Medicine. And this shows, the personalized health insights that we can get at every layer. Here, that's sort of faded off on the slide. There's longitudinal expression showing, the relative stillness of the genome and the relative variability of the metabolome and transcriptome. But we want to discuss these terms briefly before we continue. We want to discuss these briefly before we continue. What are they? The genome is the total set of genes and genetic complements. The transcriptome here are all these RNAs that get transcribed from that genome. And this is coding instructions that then go to cell assembly organelles in the body, known as ribosomes, and they get folded and processed into proteins, or the proteome, which is the total, all the proteins and isoforms and complements of those proteins. And then we have the metabolome. The metabolome are the small biological end products. They are more abundant in number. They speak to changes in the proteome, transcriptome, and genome. And because of this, are often used as the genetic, as a functional output of the genome, showing necessarily what aspects of the genome are expressed, showing what aspects of RNA are actually translated to protein, and the functionality, or for instance, enzymatic activity or the hormonal secretion of the proteome. And so the metabolome being downstream, it is the most sensitive and accurate predictor of health states. And of course, we have the microbiome, which we won't discuss today, but we have plans in Therium to develop testing here. And we have this, each microbiome, every single bacteria has its own systems biology pyramid. So as you can see, the number of data that we have to collect in order to truly map out an individual's cellular functioning at a molecular level is vast and far beyond the few dozen parameters that are returned with traditional testing, as I mentioned, the CBC, maybe a functional liver panel, OAT test, et cetera. These routine metrics are really affording you such a small, narrow view of your biology, a true appreciation, understanding of your biology, and the type of data we need to drive precision health relies on genomic, transcriptomic, proteomic, metabolomic, these omic large data's collection schemes that essentially are created to detect entire layers of your biology, or at least an appreciable sample of that layer of biology. And so,
Do you
as
mind
we
if I
see,
interject with questions here and there?
sure,
And
go
I'm
ahead.
sure you're going to answer this in just a second, but something you said was that, hey, with the current health testing model and how we approach things with the OAT test and these other tests, we're really not getting the precision that we need for early detection. So, and if you answer it in the next couple slides, great. But my question is, so what is the difference in this metabolome testing, or this metabolism testing, and this layer of the omics to what is currently available today?
Absolutely. We'll get into that when we get into the power of metabolomics in discerning Alzheimer's disease, and specifically discerning between different stages of Alzheimer's disease. And we will show that currently there is no test that can do return such a result in living humans, and do so with such accuracy as we can. And we can discuss the specificity. We're going to discuss, when we talk about accuracy, Micah, as a statistical concept, as a mathematical function, accuracy is a product of sensitivity and specificity. And we will see that later when we discuss accuracy in the terms of a receiver operating characteristic curve. And when we plot the accuracy of our panel that we found for Alzheimer's and how accurate it was in humans. So I just want to discuss here about specificity. When we return something like high cholesterol, the reason for that could be 14, 15 different items. When we return something that's, with traditional testing, you have high HbA1c, the reason for that is, again, myriad. You have low or high heme. Well, is your iron status below average? That would explain heme synthesis, for instance,
it's not
right?
very isolating.
It's
It
not
doesn't
isolating.
isolate the cause. Yeah.
In fact, hardly ever is there a biological parameter one can test, either with traditional testing or with systems biology testing, that affords one specificity. Even with metabolomics, if a metabolite, for instance, alpha-ketoglutarate, if it's high or low, if it's high, the implications are about 5 or 6 different things it could be. If it's low, there are about 3 or 4 things it could be. But the high or low levels of even one metabolite do not lend any specificity to the questions we're asking. They cannot by themselves, each one by themselves, be used to diagnose, assess, quantitate the conditions, performance, or physiology of an organ or a person. So we cannot really rely on univariate metrics. And that is what traditional testing is dependent on. In fact, it's dependent on the univariate analysis of the interpreter, usually the healthcare provider. What we see here is the number of unique models in a log function that we can have as a function. So your Y output is a number of unique models you can generate as a function of the number of variables you collect. And so we can see that 25,000, which is far less than the number of variables you would collect with a genome, or 15,000, which is right about the number of variables you would want to collect with the proteome, or even 1,000, which is somewhere between here, which is some how advanced some research-based metabolomics assays are. The number of models we can create shoots up exponentially. And why do we want to create models? Again, because each metabolite does not mean anything by itself. Each one of these parameters by themselves can indicate a vast variety of potential conditions. However, when we align certain metabolites, for instance, when we model mitochondrial health, we are necessarily looking at 5 of the 8 metabolites of the mitochondria, as well as adenosine. So we have modeled mitochondrial health using our a priori knowledge of the products and substrates in the TCA cycle. And so when we take a look at the specificity with which those 6 metabolites, 5 from the TCA cycle and adenosine, speak to mitochondrial health, the specificity is actually very high, because each one of those metabolites could indicate 1 or 2 things, 3 or 4 things, but when they are lined up together, they speak to one thing, right? It is the resolution with which we can infer to the underlying concept we're testing, such as mitochondrial health, or today, as we'll discuss, Alzheimer's disease.
So I'm sorry to be the monkey in the room per usual, but can you explain a little bit about unique models and number of variables and what that means? So as I'm understanding this graph, it's essentially saying like, hey, we are getting a very in-depth analysis of what's happening in the body. So we have all these different things that we're measuring, and the collection of those give us an exponential return on what we can see.
Absolutely.
Is that what this graph is displaying? And then
exactly. And a unique model is a model for one thing. For instance, we have for instance, let's say, for taking some of my other previous work in valley fever, which is respiratory infection. So respiratory infection or valley fever, we want to have metabolites that don't just diagnose valley fever, because oftentimes the physician is not wondering whether this person has valley fever or is perfectly healthy. Of course, this person has something. They're presenting with symptoms consistent with valley fever. However, the symptoms for valley fever are also generalized, vague, and often overlap with symptoms of flu, symptoms of bacterial and viral pneumonia. So when we say unique models, well, just to diagnose valley fever, we need 5 models, because we need to test for valley fever and then test to make sure it's not flu, it's not bacterial or viral pneumonia, it's not community acquired pneumonia. Various aspects and manifestations of similar diseases need to have models also constructed for them, so that we can narrow and pinpoint a diagnosis for valley fever, coccidioidomycosis. So this demonstrates that there's this complexity and vast potential in data analysis as these variables increase.
Yeah, I mean, absolutely, because with like the OAT test, for example, you're just getting, or a hormone panel, you're just getting an idea of like, oh, testosterone is here, but we don't have any idea of the causative agent. Whereas this, we've modeled out what are the indications of a potential disease state. And then we can say, okay, we think it's this potential disease state. However, we know these other ones have some similarities, although not identical. But then with all this data that we're collecting, we're able to rule out all the other ones and isolate which one it actually is. So it's so much more precise.
Exactly. The specificity is increased. And so even with 100 variables, Micah, we can create an astronomically high number of unique models, far exceeding human computational capabilities. Okay. So what we have established so far is that precision medicine necessarily relies on big data, and that big data cannot be computed with a human brain. So if you've come here to learn about how do I analyze the data to render an actionable insight to my patient or customer, you cannot. I cannot. No human brain can compute these models and render a cogent analysis. Any attempt otherwise would be an infantile analysis of this big data. Now, really, we have various, problems. And again, this is from our upcoming publication, which is submitted in review right now in genome medicine. And what we're looking at are these, various issues with and data analysis challenges with collation, harmonization. How do we select features? How do we find out which metabolites or genes or proteins are important for which conditions? How do we integrate them across levels. So the activity, the gene that is responsible for coding a specific enzyme. Well, we want to analyze that gene. We want to analyze the RNA that's transcribed for assembly of that protein. And then we want to analyze the levels of that enzyme and also the substrate and products of that enzyme, which are often metabolites. And so we can map our results onto the circuitry, the known circuitry of human cellular physiology, and come up with meaningful condensed health insights. And that's what we do at Therium. And this is a schema showing the analysis and computational flows and burdens of big data and systems biology analysis. And so we do all this to create personalized treatment plans, predictive analytics of that these personalized treatment plans, as we'll discuss first, show extensively and with detail what you should do, when you should do it, how you should do it, for how long, etc. The predictive analytics showing you that you have certain risks for significantly enriched disease signatures, improving diagnosis by being able to pinpoint cellular processes such as aging or cellular organelles such as mitochondria for enhanced treatment, and identify those shortcomings in a test. That is what systems biology promises, really. And so we have data integration, privacy issues, and of course this need for advanced analytics. And big data isn't just about collecting so much data. Everybody collects a lot of data. It is the sophistication and effectiveness, the accuracy and validation of those algorithms that are really important. So today we will be discussing. Yeah, go ahead, Micah.
Just to say, big data is just referring to the idea of collecting a large body of information. In this instance, through the blood, right? And so,
absolutely. Data analysis is that bridge that turns that data into knowledge, that turns that information
it needs to be
insight.
Strategic, right?
Absolutely.
It needs to be strategic in the sense that you can collect a lot of different things, whether that be in a business or what have you, your job. You can collect information on all sorts of things, but a lot of times it's not the most useful. It's finding the strategic pieces of data that you can collect a lot of those and really, really make this run well.
Sure. Yeah, exactly. And there's philosophical questions about the value of big data going forward, especially as our algorithms and computational methods become more sophisticated and more clever and are able to do more with less data. So the value of data in the future might decline, but right now we have a preponderance of data. We do not have a data problem. We have a data management crisis, really, and that's where translational research has largely defaulted on its promise to deliver the insights that the Human Genome Project really failed to deliver. And it wasn't their fault. That was an excellent initiative. It was a stepping stone and a template for science to come. It'll fuel decades, if not a century more of science. But we had a poor understanding of what the genome meant and really what it promised to show. And so we have created the Aristotle test, named after the philosopher who first committed a cogent theory of metabolism to formal writing. And we've discussed metabolomics. You saw it previously in the hierarchy of systems biology. It is the small molecular end products of the protein interactions, the RNA synthesis and translation, as well as the expression of the DNA. And so they are small molecules. They are roughly 1,500 Daltons in size. They show, as I mentioned, a direct functional readout of the physiology and show right now what is happening. Whereas for instance, the genome may show what could potentially happen, and the RNA show what is starting to maybe happen if we eventually see proteins be expressed. And while the proteome is itself a very advanced indicator, it is not as sensitive as the metabolome. And far too often, small gradual changes in these chemical signatures at the metabolite level precede changes at the protein level by years or more. And so it's really reflective. Metabolomics, as this level of systems biology, sits at the intersection between the genome and the environment, and is this direct functional readout. It is, for all the conditions that really are the greatest contributors to human death, they are heterogeneous disorders that are whose etiologies lie in the interaction between genes and environments. And so that interaction necessarily is expressed most early, most sensitively, most acutely in the metabolism. And so today we'll discuss some of my own work and how this is really done. So this is a publication back in 2021, one of my last publications of my doctoral program. I gave this talk in 2019 at the Arizona Physiological Society here over here in Tempe. And this issue is we're looking at metabolic profiling in a sample of male and female individuals, 48 individuals post-mortem, taking a slice of their prefrontal cortex, looking at normal controls, high pathology controls, mild cognitive impairment, Alzheimer's disease. Of course, we go to the lab and perform our extraction protocols and return those metabolites. In this study, we used GCMS and a combination of LCMS. And so this is actually the Aristotle test. So the test that we offer, the actual assay with the actual metabolites that we offer, was run in this study. And so we go through a process of statistical analysis, how we identify biomarkers, and then we look at pathway effects and enzyme effects. And then we offer a biological interpretation of what's happening at the cellular level that is leading to the pathologies and symptomologies that are associated with high pathology individuals, mild cognitive impairment, and those with Alzheimer's disease. And so what we found were 4 metabolites who were significantly changed between groups. So here's levels of lauric acid. You see a relatively linear decline in lauric acid as you go from normal controls to high pathology controls, those with mild cognitive impairment and Alzheimer's disease. Myristic acid, we see a slightly different relationship, which again can be modeled. Same here with stearic acid. As progression increases, we see significant changes in these levels. Again, with palmitic acid, which is a very much a pro-inflammatory fatty acid, we see, again, characteristic changes, all significantly different between these groups using a MANOVA test with least significant difference correction for false positives. And what we're seeing here are that we have 4 metabolites that let us assess an individual. And again, each one of them may only assess one aspect. By themselves, they may not be that accurate. So we go through various feature selections, and we haven't gone through everything. We haven't gone through everything right now that we did in that study, just the highlights. So we did some feature selection to narrow down these 4 metabolites, because we had more metabolites that were significant, but these were the ones we chose to model. And what we see here is a random forest classification.
Real quick question on that.
Go ahead.
Might help me personally in clarifying. Can you go back one slide?
Sure.
So this goes kind of back to my question on the process of elimination, right? So we're looking at these different groupings. So I see under like stearic acid, the mild cognitive impairment and the Alzheimer's disease have a pretty similar grouping. So
those are not significant. Those are not significantly different from each other. So you see why we need more than one metabolite here, Micah.
That's exactly what I'm getting to.
Yes,
Yep.
yes, because these are not significant. So how do we discern? We really need to be able to discern, normal controls from everything else. Well, we can do that actually using myristic acid. And actually, there's no significant difference between normal control and high pathology controls here either. And so we need the differences between like normal control and AD in myristic acid to tell normal control apart from AD. So we are really making 1, 2, 3, 4, 5, 6. We need, up to 6 metabolites. Depending on the performance of these metabolites, we need up to 6 metabolites to discern 4 different stages of Alzheimer's, because it's not just Alzheimer's and controls, right? That can maybe be done with even 1 metabolite. But when we talk about levels of 4 metabolites, we need discernment between normal controls and high pathology controls, discernment between normal controls, mild cognitive impairment. Every combination needs to have a metabolite that can be modeled so that we can then do an overall modeling and show that with every single one of these metabolites, we have, for random forest analysis with 500 decision trees, this is a form of AI, this is a form of machine learning algorithm that takes the data and the expression in these subjects, and across 500 decision trees computes an out-of-bag error. And then we take it and we plot it. What do we see here? We see sensitivity on the y-axis. We see specificity On the x-axis, I discussed how we were going to show this earlier today. This is the true positive rate. This is the false positive rate. You can think about sensitivity and specificity in those statistical terms. And when we plot the sensitivity and specificity, we get an area under the curve of 91.7%. So 91.7% of our individuals in this study were correctly classified using levels of these 4 metabolites. And of course, these 4 metabolites coming from GCMS platform are also available on the Aristotle test. Now, I mentioned in the overall schema of the study that we can also then take all of those metabolites that we analyzed. In this study, we analyzed more than 126 that's available because we use another platform as well in conjunction, LC tandem mass spec. And so we can take all of those metabolites and their relative levels and map them to the known human circuitry. We can map them to the human metabolome, just like you would take your genomic results and map them to the genome. We are mapping your metabolite levels to the metabolome. And what we're seeing here are significant changes in arginine and protein metabolism, linoleic acid metabolism, coupled with significant changes in lysine degradation and fatty acid metabolism, where on the bottom we see pathway impact, which is a weighted expression of enrichment, and on the y-axis, we see negative log P, the significance of them. So higher pathways are more significantly impacted, and those to the right have a greater change. And those are different, actually, significance and magnitude of change. And so, Micah, do you have a question?
Yeah. Oh, just a quick note. We're 33 minutes in, so
we're
go
almost
relatively fast. I know you're. I hate to speed you up, and I caused some of the delay, but just wanted to.
No worries. We're gonna go through. This is just to give you an idea that we can then take those same metabolite levels. We did it for pathways. We're going to now map them to enzymes, to the products and substrates of enzymes. And what we can see here is a high degree of enrichment. We see it in network view, a high degree of enrichment in mitochondrial enzymes. When we look at this biological interpretation, now what we're seeing here is that with an increased pathology related to AD, we're seeing gradual changes in the fuel that cell decides to use. We're seeing changes in substrate utilization going from normal glycolysis to degradation of branched chain amino acids, and then finally beta oxidation of fatty acids. And these are all, again, using our large data collection coupled with sophisticated data analysis. So this is how we're doing it. Again, we are processing these tissues. We're using an assay that is incredibly similar, and in fact, identical as far as its GC parameters to the current Aristotle test. And with our biomarker analysis and advanced bioinformatics, we are able to infer metabolites that speak to the generation and aggregation of amyloid beta and tau protein in the context of Alzheimer's disease. So that is essentially what we do. Like as Micah mentioned, there's a sample collection, there's a GCMS, and there's a data analysis stage of every test. It renders 126 metabolites. These span 68% of all known human metabolic pathways and probe 14 different chemical subclasses. And to date, it represents the most advanced metabolomics test, but as far as we know, the most advanced metabolic test on the market. This is in our actual lab. This is the metabolites. This is the actual machine we use, the instrument we use to separate and quantitate metabolite levels from our dry blood spot samples. This is the same machine that was actually used in those studies that we went over. And we're going to go over one last study here. So now that we have this ability, I've shown you that we have this ability to detect different stages of Alzheimer's disease. We also have the ability to simulate treatments and, again, give you a personalized precision protocol to follow. I published this middle of last year. This was with excellent researchers at the Banner Sun Health Research Institute, as well as Barrow Neurological Institute here in Phoenix. I was working on this during my postdoc. We managed to publish this in Active Neuropathologica, world-class journal. And really, the reason was we had advanced data collection like metabolomics, coupled with multi-model experiments. So we did experiments in mice, for instance. So you can see here that we have changes in these mice. We have a triple transgenic model of mouse, which is designed to develop Alzheimer's disease. And what we have here in the graphs on B We have those without choline, so choline CH minus, and then we have those that are non-triple transgenic. So this is showing the model and its propensity to develop Alzheimer's disease. What we see here is a decrease in cortex choline for those mice that are triple transgenic. We see here that choline, cortex choline, is significantly correlated with plasma choline. We see the R squared, we see the R correlation and the significance value there. We see that plasma choline is significantly impacted with acetylcholine, obviously, as one is a derivative of the other. Plasma choline is significantly correlated with TNF alpha. We see, again, cortex choline significantly reflected in plasma choline and vice versa. And so this is showing that in the mouse model, choline is significantly associated with the development of Alzheimer's disease in these models. Now, when we look at humans, so these are those who have mild cognitive impairment. We saw this same cohort in my previous study. And what we're seeing here is that choline is significantly correlated with acetylcholine, both in terms between those who are controlled, have no evidence of disease, as well as those who have a small amount of mild cognitive impairment and those with high mild cognitive impairment. We're seeing significant changes here. We're seeing a significant change between choline, for instance, and neuritic plaque density, which is, again, and this is in humans now, not in mice. So in our human samples, we're seeing choline is significantly associated with neuritic plaque density, inversely associated. So more choline, less neuritic plaque density. We're showing here, again, for instance, with choline, as choline increases, Braak stage decreases. So the pathology staging for these individuals actually goes down. When we see choline go higher, we see no change in global cognitive score. So it's showing us that choline is mostly associated with the pathology of Alzheimer's in these individuals. Now, those were mild cognitive impairment individuals. What about choline's associations with Alzheimer's disease, both those with moderate Alzheimer's and those with severe Alzheimer's? Again, significant, almost significant association between choline and global cognitive score. We see significant decreases as choline goes up, decreases in neuritic plaque density. We see significant increases in choline are accompanied by significant decreases in Braak stage. The same thing is true with brain weight. So as choline goes up, we see a significant increase in brain weight. So the choline is in many ways, it seems, protective of neurodegeneration and loss of brain mass. And so we have various levels in micromolar concentrations of brain weight and in micromolar concentrations of choline, and we have their known association with Alzheimer's staging. We have their known association with the effects of brain weight, for the effects of Braak stage, for the effects of neuritic plaque density. Everything that we can mine from this article, we have also now looked at significant associations between our metabolites and the acetylcholine and choline levels in humans. And what we see is a heat map on top showing the choline and acetylcholine levels. And then we see 4 metabolites. These are also 4 metabolites of the Aristotle test that are significantly associated with these 3 groups: valine, hydroxyphenylpyruvic acid, NMA, and ferulic acid. And what we see here, again, is a significant association. So given levels of these 4 metabolites, we can actually infer your levels of choline. And given any marked signature or increased risk of Alzheimer's viewed in the metabolome, we can prescribe very specific dosages and durations of choline, for instance, to then supplement, augment, or counteract, or hopefully even turn back the time on a lot of these pathologies that are consistent with the development of Alzheimer's. And so that is bringing us to our next topic, which is the role of digital twins in healthcare. So you saw a very small example just now of what can be done. We can deploy very comprehensive data collection platforms to analyze a vast amount of data in individuals. We can then couple that with advanced bioinformatics to render very sensitive and specific health insights on an individual. We saw that in the case of Alzheimer's disease, but that can be extrapolated to any number of health conditions. And then we saw how Alzheimer's disease, the progression of Alzheimer's disease as a pathology, is related to metabolites, and how those metabolites can be used to assess the levels of choline in an individual. And from that inference, then recommend, based upon the levels of these metabolites, how much choline an individual should supplement with in order to decrease the risk of Alzheimer's or any risk that was noted. Now we're going to digital twins, and this is a tool that is increasingly popular and will enable doctors and researchers to visualize these complex interactions. It is a platform by which they simulate the effects of these on an individual, and it serves as a predictive tool. And that's what we do at Therium. So we will, yeah.
Interject one question that's right on this topic, I think, but Garrett Athenas asked, how can the data be correlated to a treatment plan that has the same specificity and sensitivity as the data provides?
Absolutely. You're absolutely right. So the treatment plan is necessarily contingent upon the data it's trained on. And so even with something such as choline, we didn't just feed it our study. In fact, our studies represent, most of our studies aren't even indexed because some of them we feel aren't really up to snuff. Some are, for instance, this choline, but even this inference to choline supplementation is trained on a vast number of studies that have shown similar results across different populations and across different models. Even cellular results can help inform this. So you're never going to get 100% accuracy. In fact, if you ever see a receiver operating characteristic curve with 100%, you should be very suspicious. Our goal as scientists is to get close to 90% and then validate that model. So it's really important that we're looking for things such as overfitting. So you mentioned, the sensitivity and specificity of that model is really contingent on whatever was reported. But not only do we report on more than one study for such an intervention, but we validate these models such that we check for overfitting. We make sure that the projections that we have are not too closely fit to the training data and therefore are not generalizable to new cases or new customers as their data is coming in and analyzed. And that really is the trick. So you're never going to get perfect sensitivity. You're never going to get perfect specificity. You can get very, very close, far beyond conventional measures. And by leaning on not just one study or a set of studies from a particular research group, but leaning on the best studies from across the science, we can increase and sort of average out our sensitivities and specificities. We can check for model overfitting and making sure that we're not relying on too specific of a signature that is just really idiosyncratic and belongs to a certain sample. It's a function of sample error. And those are the ways that we can really assess these health gaps that you see and use the differences there to inform simulations. And what we do, for instance, in the case of choline, would be to take that choline supplementation, the various studies that have shown different levels of supplementation, and simulate the effects of that on that individual's digital twin, on that virtual health representation, and simulate the effects of that 1,000 times with 15% randomness built in. So we change things a little bit, 15% every iteration. And the idea, therefore, is to emulate real life and to see across those 1,000 iterations which interventions on average and which dosages and what quantities taken at what time of day were predicted to yield the greatest increase and close this health gap the quickest for that individual. That is the computational process behind it, and that's how we deal with limitations that are inherent in these translational efforts.
And Garrett has a follow-up question that's
Sure.
on the subject as well. So are the clients or patients sending usage of these integrations back to then see what is the best correlated to the efficacy of the recommendations provided?
So if your question is whether or not, for instance, we send the raw statistical outputs of an analysis to customers, then no, that's not what we do. For certain practitioners on our practitioner platform, we have provided certain, very nuanced analysis and bespoke analysis to answer some questions they may have had. But if your question is to see then at retest how many of those were let's say we give a protocol with 15 recommendations, and they're very specific. This is much exercise, this much of this food, this supplementation, change, your living habits this way. If what we do is then we assess compliance at retest. So every domain that we test comes with a retest recommendation as well as a composite recommendation for retesting. And the idea is that based on the science and what's been reported, those interventions that have been reported to affect those metabolites, there's a window of time in which we would necessarily expect to see changes of those interventions reflected in the metabolism. And so we benchmark that against the science as well. And so let's say somebody has a retest recommendation of 4 months. They come back to us in 4 months, they retest and to see if their scores went up or down, if their disease signatures enlarged or augmented or diminished. And we assess their compliance and their observation of the protocol. We assign a quantitative measure to that, and therefore, when we do this digital twinning simulation again at retest, and we close that data feedback loop, it actually becomes far more refined and precise for that individual, because now we don't just have an estimate of how this individual will respond to a specific intervention given similar responses of people that are similar to that individual, but now we know a relative change, a delta that lets us really estimate with far more granularity how this individual is different from individuals like them and how they respond and to what they're predicted to respond to. So every iteration of this retesting, it actually, and that's the beauty of digital twinning as a computational endeavor, is that at retest and at every input of new data over time, this becomes more accurate, more predictable, and more efficacious in terms of patient outcome.
Answered my question. Hopefully it did for Garrett as well. Are we about done with the slide set here, so we can go into, because I have a couple things to say as well, and then get into Q&A, and I'll be 60 seconds, but.
And that's it. I mean, that is the end of the presentation. It was really just a and we can discuss what kind of recommendations do we give and how granular is it, and will I be able to understand the recommendations? We can go over all of those, but that is the long and short of the presentation today.
Cool. Yeah, guys. So just a really quick recap. So there's the different layers of systems biology that are looking at different parts of the body and how they're working and what's going on. The genome might give specific insights into what's happening genetically, but it has limitations. And one of the reasons that Dr. Jasby loves the metabolism is it's so dynamic. We can see when you take maybe not when, but we can see if you take ibuprofen or caffeine and what the intake of those things are. So it gets really sensitive into these different aspects of what's happening in the day-to-day life. And so with that increase in sensitivity and all the things that we can see, we can pull, we can pull a huge amount of data together, and we can translate that through, like he was talking about, with the process of elimination and all these other things, like the choline being modeled for different aspects of how the brain is performing and the effects that it might have there, and get a really good understanding of what's happening in an individual. So as clinicians, this can be really helpful for really bringing personalized or precision medicine to your patients, and not only improving their outcomes, but it does have a substantial revenue boost for most clinics, in that, if people are seeing third-party data by good science, like somebody like Dr. Jasby is providing, that's pretty incentivizing to say, yeah, okay, well, there's this analysis that was done on my blood that tells me I should probably take X, Y, or Z supplement, that I should probably institute this intervention or this, at times, not super often, it'll even recommend a drug, but it'll produce lots of different recommendations for people, and they're much more likely to comply with that when they have third-party data like this. So if you guys, there's a couple of options. We're going to get into Q&A right now, so it's not over, but if you guys want to work with us as a clinic, and consumers can go to the website and buy that, that's great. That's www.therio.me, which you can see on the screen behind Dr. Jasby. And I'll put it into the chat here. But as far as working with us as a clinic, the best way is either drop your email in the chat right now, and I'll save that. I'll send you an email afterwards, or you can send me an email directly to Micah, and I'll at therio.me, and I'll put that in the chat as well. But I help clinics get onboarded with this process and bringing it into their clinic, making sure it's successful, making sure that it's profitable for you guys, and and all that kind of thing. So without further ado, guys, feel free to fire away questions. Feel free to turn your camera and microphone on, as well as if you want to. You're more than welcome to do that. So I had just enabled that feature, and you are now welcome to unmute and ask questions away. I know Dr. Jasby usually has a big waterfall of information and goes into all sorts of things. I swear, every time we do a talk, you have a totally different slide set too. You don't reuse. You
I'm not a
really
fan of
think about the
reusing.
subject and
Yeah.
develop something totally new. So, hey Garrett, how's it
So,
going, man? Looks like you're muted in case you were wanting to ask a question. I see your camera did turn on though. Are you able to unmute yourself?
there it
Okay,
is.
perfect.
Yes, I can. Thank you.
How's it going?
Oh, great. Great introduction there. It's definitely groundbreaking and eye-opening to what the potential is out there for this.
Sure. Thank you.
Yeah, absolutely. And I actually have a call with your brother in
Yeah.
a little over 30 minutes here. So you're welcome to join that as well if you'd like.
Yeah, his background is a little more in the I guess the mental health kind of aspect, but I think this definitely, as you say, Alzheimer's, I think this definitely correlates to, obviously the serotonin, dopamine, COMT levels, things like that metabolically, you can definitely work with, especially the microbiome wise. So I think there's
Yeah.
definitely some strength that this can provide. I don't think that's really out there.
And ultimately, Garrett, we do have plans to develop an entire ecosystem of systems biology testing later on this year. As long as we meet our growth targets, we should be launching a microbiome test of the stool, and then after that will be proteomics and then transcriptomics and the genome. Although, I of course I'm a metabolomic scientist, the genome is an excellent thing to know, and once you have all these other layers of systems level information, the genome adds this final resolution piece, whereby you understand what is coming directly from the host, and for instance, what is coming from the outside, or what is being expressed from interaction with microbiota. And so the genome is essential. And as I mentioned, it really was the template for which decades, if not a century or more, of science has been and will be built upon. But we've gone past since because of our curiosity and the need to. The genome has proved inefficacious in providing, for instance, in Alzheimer's, really you're looking at the testing of like ApoE4. In the case of breast cancer, you're looking at BRCA1 or BRCA2 genes, right? And so these are really asserting broad probabilities. You may have a 20 to 80% chance of developing breast cancer. Well, okay, is it 20 or is it 80? And so that is really the problem with genomics, and of course the fact that it doesn't change and it's not temporally dependent. It can't really be used as a therapeutic monitoring tool in the same way that the other layers can. So every layer has drawbacks and pros, but keep in mind as we grow, our goal is to provide this deep molecular fingerprinting of an individual. We started with the metabolome again because of its sensitivity and specificity, but we will grow, and I hope that you see that and can benefit on it.
I guess from my experience in education, the genome helps, but it might help a why, but doesn't mean metabolically it's expressing. So I've taken all these courses, and there's so many companies that will take this gene test and we'll give you XYZ and you have to change these things. And I just feel like it's fallen so far short to be
Absolutely.
applicable. It's like, well, maybe there's lower, expression, maybe it's higher, but it says maybe, and it's like, there's too much maybe. I think metabolomics just says, well, this is
This is
what's going
what's
on.
happening right
You
now, and it
could
can change.
go backwards.
Yeah.
You could say, well, this gene's probably doing this, but I'd rather take metabolomics and go, well, this is what's going on. I'm going to add these kind of integrations and see what the results change, and then keep using that data. I think is way more functional.
Absolutely. I think there was somebody asked about SNPs testing. Is that you, Garrett?
That was Garrett.
Yeah,
He's the only one that's put questions in so far, by the way. So feel free to ask questions if you have any.
Feel free to ask anybody else. Yeah, absolutely. But for SNPs, for instance, we can go backwards, like you mentioned. We can look at the metabolites and we can infer, given their levels, we can infer the significant presence or absence of SNPs. And in fact, for one of our health testing domains, the mutational load, it's a combination of various nucleoside and nucleotide metabolites combined with an enrichment analysis of over 4,598 different SNPs, of which we take the proportion for which you're significantly enriched for and calculate a score based upon those and the levels of those metabolites I previously mentioned. But SNP testing, for instance, so the genome does not speak to the metabolome, right? But the metabolome can speak to the genome. All layers can always speak to the layers above them, but never below. It is sort of like, 2 goes into 4, but 4 doesn't go into 2. It really is a common denominator issue. And so with the genome, we get a very limited view. It's necessary, and nothing else can really take place without it. Molecular studies are informed by the human genome, but ultimately it's incomplete for the needs of health testing for most conditions. Again, if you have something like Huntington's, well, genetic test of Huntington's won't necessarily tell you if you will develop and ultimately die of Huntington's. However, for most conditions that are the greatest contributors to human morbidity and mortality, it's not a gene or a set of genes or even a set of dozen genes that can provide that accuracy that, for instance, 4 metabolites can in the context.
Now, would this be correlated to more like the epigenetic data as the metabolome, like the expressional data relative to the genes? Because there's genes that are like, obviously there's transcription and proteins and goes on and on, but there's only so many things or so many genes, from my understanding, that you could really on an epigenetic wise actually have a endogenous interaction with, right?
Yeah, absolutely. So metabolites are more varied, and they're product of both endogenous synthesis as well as interactions with the environment. But yeah, you are right in the sense that metabolomics is more tightly correlated to transcriptomics rather than, or the epigenetic profile, the epigenetic expression, than it is the genome. Yes, absolutely. Yeah. Somebody did ask about A1C. We don't currently test for A1C. That is a protein, and so it's too big, its usefulness is really limited to a few disorders, predominantly of.
Well, let's start with the disorders then. What are the disorders that A1C has a concern with, and can we look at those?
We can, for instance, much more accurately and sensitively. So when we say A1C, we're looking at, relatively like, 3-month snapshot of your glucose metabolism. Well, we can look at your glycemic response. We can look at insulin signaling. We can look at glucose uptake and insulin sensitivity. All of these processes can be looked at using these metabolites. For instance, valine, leucine, isoleucine, which are in our panel, were displayed in the energy shift that accompanied the gradual progression of Alzheimer's disease. We can look at that, and in fact, Alzheimer's disease is known as type 3 diabetes in some cases. In some circles, people call it type 3 diabetes, and so it's very interesting. And we don't look at A1C, but we can look at the processes that A1C reports on. We will ultimately include A1C. It is a good parameter. It is incomplete as any parameter by itself, but it's a good parameter. It's in the proteome. We will get to it eventually, and in the meantime, we are testing the processes that it influences using, the metabolites involved therein.
So is it primarily just looking at diabetes when you're testing for A1C?
Mostly, yeah. There's some other things it can detect. For instance, it increases your risk of cardiovascular event as well. So it can be used as a sort of assessment tool for your physician to look at your relative risk of cardiovascular disease. Somebody did mention, and I only have a minute to go because I do have another meeting that starts right now. Somebody did mention, how can it be used for people with Lyme? Well, it can not only be used to detect the presence or absence of infectious diseases like Lyme disease, but it can also be used to assess the effects of that pathogenic process on aging, on the mitochondria, on cardiovascular health, liver health, your inflammation. For instance, inflammatory score is something that we calculate with the Aristotle test and have a very good indicator of that beyond traditional inflammatory profiles and panels. And so for people like Lyme, not only can it really corroborate the presence or absence of infection, such as Lyme, but it can look at the effects of that disease, such as Lyme, on important health domains, and as well with retesting be used as a therapeutic monitoring tool to assess the efficacy of those interventions. Unfortunately, I do have to go, Micah.
Yeah, no worries. Sorry. I'll wrap up from here. So I just want to say thank you to everybody for joining today. It's a real pleasure to speak with you. If you have any questions, anything I can help with, please just email me, micah@therio.me. I'm going to put it in the chat one last time. You can take off, Denise.
Sounds good. Thank you, guys.
And then if you want to buy a test directly, you can go to the website, which is therio.me. But we're here to serve you guys. We want to give a good understanding on what precision medicine is, personalized medicine is, and how these things essentially equip you to get better outcomes and to change lives. At least that's why I'm in this. So again, thanks everybody. We'll see you again soon. Bye.